Yes, so the next presentation is titled Presentation of a Decentralized System for Nutrient Recovery from Human Urine. Professor Oladoja, good morning, Professor, is the chair,
Good morning.
Hydrochemistry Research Laboratory, Department of Chemical Sciences, Adekunle Ajasin University in Nigeria. His expertise in environmental interfacial chemistry has been deployed to produce interfacial engineered filter materials and green coagulants for water and wastewater treatment. His works on aquatic ecological engineering has been employed for the restoration of the eutrophicated aquatic system and development of innovative strategies for resource recovery from wastewater. Take it away, Professor.
Thank you. Good morning from China, Beijing specifically. I'm trying to share my screen.
Yes, you should have screen sharing abilities as a co-host. Let me know if that's not showing up for you.
I'm trying to share my screen.
You should have the ability to share your screen. Do you see the share button in the bottom?
Yeah.
Looks like it's working.
Yeah. Can you see my screen?
Yeah, we can see your screen.
Okay. I'm Nuruddin Abiola Oladoja. I'm making this presentation on behalf of my other colleagues that is listed here. Can you hear me?
We can hear you, but it's not in presenter mode. We see your whole PowerPoint presentation with the notes. Are you able to put it in presenter mode?
The presentation mode.
I think in the bottom right, the far right icon on that very bottom row there, just before the Zoom. Or if you click on the slideshow tab at the very top. That's another way there.
Or we could just go with it like it is.
Yeah, we can see it pretty well this way.
We can see it.
Yeah.
See, okay now. Okay. The title of our presentation is Preparation of a Decentralized System for Nutrient Recovery from Human Urine. It is incontrovertible that man produces about 1.5 to 2 liters of urine per day. And this urine, which is known as yellow water, the human urine, which is known as yellow water, it is very high in nutrients, specifically phosphorus and nitrogen. A comparison of the nutrient fraction of human waste, which comprises of urine and feces, with the synthetic fertilizer, which is the conventional mode of fertilizing our plants, is shown on the screen. We can see that the nutrient fraction is comparable with the commercial fertilizer. But the question is this: despite the rich nutrient fraction of urine, why is it not being used as we are using the conventional fertilizer? So we look at some of the factors that responsible for our inability to use to conventionally use urine or human waste as fertilizer. Some of the factors identified consist of high cost of transportation of large volume, because a large volume of urine is generated by women. So the high cost of transportation, the unpleasant smell, the risk of pathogen and pharmaceutical residues, and the need for long storage time and large storage volume for sanitization, because sometimes urine is produced, but it is not required for usage at the same time. So because of that, you may need to store for a long time. So considering the peculiarity of this, nutrient resource in terms of ethical and cultural values, especially in the developed world, we are of the opinion that a decentralized system is required to capture the nutrient fraction of urine. By doing this, we'll be able, to manage some of the challenges that are associated with, the direct use of yellow water. So our research aim was to develop a decentralized modular system for the use of homeowner and small-scale business outfit for the recovery of nutrients from yellow water and reuse of the recovered nutrients for sustainable agriculture. So our objectives includes evaluation of the nutrient recovery and pollution remediation potential of gastropod shell in synthetic feed and real yellow water. Then we determine the operational parameters for the use of gastropod shell in the modular reactor, which is the decentralized system. And we characterize, in terms of nutrient assemblage, elemental composition, and the safety evaluation of the nutrient-laden gastropod shell. Then we determine the potential of the recovered nutrients as fertilizers in plant cultivation. So one of the questions that I feel should agitate our mind is the choice of our reactive material, that is the gastropod shell. Why did we choose gastropod shell and not other material? Gastropod shell, based on our previous study, we found that we characterized the elemental and mineralogical assemblage of gastropod shell, and we found that it is a calcium-rich material. And based on basic, chemistry or chemical knowledge, we found that the calcium-rich material has affinity for phosphorus. And this has been proven in a number of studies that we have conducted prior to this, and which we have reported in Oladoja et al. 2012, 2014, 2016, and 2017. And we also observed, based on our studies, we found that the mechanism of phosphorus capture by gastropod shell was through the crystallization or precipitation of phosphate minerals. And we have of the opinion that if phosphorus can be removed via phosphorus mineral crystallization, then the other fraction, which is the nitrogen fraction, can also be occluded into, the fossil mineral during the crystallization process. In addition, we observed that one of the challenges with urine, especially during storage, is the loss of the nitrogen fraction through urea hydrolysis. And it has been proven that the extreme of pH can hinder or inhibit the hydrolysis of urea, thereby preventing the loss of the nitrogen fraction. And from our studies, we found out that gastropod shell in water increases pH of water to the extreme alkalinity. Thereby, we believe that because of the extreme alkalinity of the effect of the gastropod shell, it will inhibit the urea loss, thereby preventing the nitrogen loss from the yellow water. So how did we carry out these studies? How did we achieve the aim of our research? We started from preparation of the reactive material, that's the raw gastropod shell and calcined gastropod shell. We worked with both the raw and calcined gastropod shell based on our previous study that shows that thermal treatment of the shell enhances the potential for nutrient capture. And we also prepared the synthetic yellow water based on the method that is used by the medical students for the preparation of synthetic urine. Then after, we carry out the determination of the rate of nutrient removal and determine the equilibrium isotherm to be able to derive some operational parameter for the nutrient recovery process. Then we carried out the batch-wise nutrient recovery from human urine, from real human urine, and we characterized the recovered nutrient, looking at the surface architecture, the elemental composition, and the mineralogical assemblage. Then we derived the operational parameter from a column reactor, which is similitude of real life situation. So we designed a modular column reactor, which is the decentralized system, taking into consideration different hydraulic residence time in the column reactor by packing a column with the granular, raw, and calcined gastropod shell. And this was, operated at different hydraulic residence time of 6, 12, and 24 hours. Then after, we determine the nutrient speciation in the recovered nutrient and the safety risk. Then we evaluated the potential of the recovered nutrient as fertilizer in plant cultivation. Our findings. The graph on our screen shows the time concentration profile of phosphorus recovery in using thermally treated, which is denoted as CGS, and the raw gastropod shell, which is denoted as the GS. From the graph, we can see that the phosphorus removal capacity of the thermally treated gastropod shell was faster, the rate was faster, and the capacity, the magnitude of phosphorus removal was higher than that of the raw gastropod shell. Then for the nitrate removal, because we monitor both the total nitrogen and total phosphorus, for the nitrate removal, both the thermally treated gastropod shell and the raw gastropod shell showed similar capacity in terms of removal efficiency and the rate of removal. On our screen is a summary of our kinetic study for the determination of the rate of the removal. We found that for the total phosphorus, the equilibrium time for phosphorus removal was achieved at less than 30 minutes. It was very fast. Whereas for the raw gastropod shell, it was around 120 minutes. And for the two reactive material, we fitted the removal process, the time concentration profile, to some kinetic equation. We found out the pseudo-second order equation gave the best fit for the total phosphorus recovery, which is an indication of occurrence of chemisoption. That is the reaction between the phosphorus and the reactive material. That is both the thermally treated gastropod shell and the raw gastropod shell. And for the total phosphorus removal, the rate for the thermally treated range between a different dosage, range between 1.06 to 8.57, whereas for gastropod shell, raw gastropod shell, it range between 0.044 to 0.00, which shows that the rate in the thermally treated was higher and faster than that of the raw. And then for the for the magnitude of total phosphorus removed, for the thermally treated, it ranged between 76.92 to 270.27, whereas for the raw, it ranged between 4.68 to 34, which shows that for the thermally treated, the thermally treated gastropod shell had higher magnitude of phosphorus removal. Both reactive material, that is both the raw and the thermally treated, showed poor affinity towards nitrate removal, but it showed better affinity for phosphorus. And the value of QE for nitrate recovery was comparable for the two, we can see on the screen. Then, because of the nature of the mechanism, the underlying mechanism of the phosphorus removal, we also fitted our data, the time concentration profile, to have Vrame fractional kinetic equation, which fitted better than the pseudo first order, but it is not as good at the chemisoption. Then, for the equilibrium isotherm parameters, we can see that for the thermally treated, it's fitted for the thermally treated and the raw, it's both fitted to Freundlich isotherm equation, which is an indication that the surface of the material, both the thermally treated and the raw, they have an heterogeneity site for adsorption. And the monolayer Langmuir adsorption for for both thermally treated and the raw. They are shown. For the thermally treated, we had a monolayer adsorption capacity of 294.12 for thermally treated and 89.29 for which, affirmed what we obtained in the kinetic experiment. And then for the nitrate, Freundlich isotherm fitted for the thermally treated, whereas Langmuir fitted for the raw sample. And the monolayer adsorption capacity for nitrate adsorption was low and comparable for the two reactive material. Then when we carried out the pilot study, batch-wise pilot study, batch-wide nutrient recovery using real human urine, the physicochemical, we looked at two issues here. We compare the physicochemical parameters of the raw and the treated. And from the table on our screen, we highlighted some portion of the result. We can see the total phosphorus removal. We had the initial phosphorus removal was 14 for the raw urine, 14,864. No, the total nitrogen, sorry, was 14,864. And for the treated, we found that the residual treated was still very high, 14,429, which shows minimal removal. Whereas for the total phosphorus, it was 498.75, and it reduced to 15.07, which is more than 90% removal. Another very, very important parameter that we look at was the COD. Because of the biological fraction that is contained, apart from the nutrient, human urine contains some other biological molecules, so which we believed can contaminate or can also be removed by the process. But we found that there's a minimal reduction in the COD value. We had 5,788, which reduced to 4,529. With this, we believe there's minimal contamination. There will be minimal contamination of the recovered nutrients, which make it safe for agricultural use. Then the surface architecture of the recovered nutrient is shown above, and the elemental composition analyzed by EDX shows the presence of some elements and that are present. Then the mineralogical assemblage analyzed using the SRD, X-ray diffractometer, shows the presence of two major phosphate minerals, which is whitlockite and apatite, which we're going to discuss later in the distance. Then the phosphorus speciation, that is the phosphorus speciation, shows the presence of calcium phosphate and hydroxy apatite. The different fractions obtained are shown on our screen. Then in the modular reactor, which is the decentralized system, they conducted at different hydraulic residence time. We found out that the concentration of the nutrient fractions in the effluent, that is in the treated urine, varies with time and with the hydraulic residence time. And the volume of yellow water that was treated was also hydraulic residence time dependent. And the value reduced from, we have the highest value at 6 hours resident time, and we treated about 13,080 ml of urine of the yellow water at 6 hours resident time. Whereas, when we increase the resident time to 24 hours, we had about 5,880. Then the total phosphorus adsorbed, that is the total phosphorus taken up by the bed, by the gastropod bed, was 1,497. It ranged between 1,497 to 2,279 at the different bed height as we increased the bed height from 6 hours to 24 hours, which was far greater than that of the total nitrogen. We obtained similar results in the batch adsorption studies. And then we found out that irrespective of the influent hydraulic residence time, that is, irrespective whether it was the hydraulic residence time was 6 hours, 12 hours, or 24 hours, we found out the temporal value of the physicochemical parameters like the pH, electrical conductivity, and total dissolved solid were similar for all the columns. Then for the pilot study, we conducted our pilot studies using the best using the best hydraulic residence time, which was 12 hours. And the results are presented, which shows, the effluent phosphorus concentration, the nitrogen phosphorus concentration, that is the total phosphorus and the total nitrogen, and some of the parameters. They are shown on our screen. And we found out that the Km value, because we chose the 12 hours residence time, because the Q mass, which is the total amount of, was the total amount of nutrient taken up or absorbed by the column, was the best for the 12 hours period. And we found out that relative to the synthetic yellow water, a diminution in the nutrient fraction recovery efficiency of the reactor was observed. And this was attributed to maybe the presence of some other biological molecules that impeded the absorption efficiency. Then the volume of water treated, the value of the Q mass, that is the amount taken up, and the nutrient fraction absorption yield were lower for both nutrient fractions in the real life system than in the synthetic yellow water. And the reactor showed better recovery efficiency for total phosphorus fraction, which was 2,617 than the total nitrogen capture. Then the safety risk potential, determination of the safety risk potential of the recovered nutrient. The gastropod shell column exhibited varying degree of antibacterial activity against all the bacteria isolated from the yellow water. And at the minimum inhibitory concentration, that is the MIC, the growth of all the bacteria was completely inhibited, which makes the recovered nutrient safe for agricultural application. Then we evaluated the fertilizer potential of the recovered nutrient with maize and tomato cultivation. And from our screen, we can see this is for the maize planted with urine, this is for the control, and this is the one planted with NPK, which is the NPK, which is the conventional fertilizer. Initially, the NPK fertilizer behaved, we did same with tomato. This is for the control, this is for the urine planted tomato, and this is for the NPK. The ability of the recovered nutrient to substitute for NPK fertilizer was established. Initially, for the first 15 days, we had, the NPK fertilizer behaved or performed better. That is, the NPK fertilizer planted cross performed better than the recovered nutrient planted plant. But after the 17th day, we found out that the recovered nutrient plant became better than the NPK fertilizer, which is an indication that the recovered nutrient acted as slow release fertilizer. And we observed that the nutrient concentration in plant grown because we harvested the biomass and determined the nutrient fractions of the biomass. We found that it was significantly higher than the control and comparable with that of the NPK. And in addition, we found that the recovered nutrient also boosted the soil pH value, which is beneficial to acidic soil. Conclusion: the total phosphorus recovery efficiency was far higher than that of total nitrogen, but the total nitrogen recovery was the rate limiting step because it was a slow step. It was faster. The rate of total phosphorus was faster than that of total nitrogen. So the total nitrogen was the rate limiting step. Then the hydrolysis of the urea fraction was hindered, thereby preventing the nitrate fraction loss. That is, the process hindered the hydrolysis of the urea fraction into the respective components. So at least that prevents one of the, that was able to mitigate against one of the challenges of using urine. Then the selectivity was much higher, and it showed preference for the nutrient than other fraction of the urine. And the nutrient efficiency of the modular reactor was hydraulic residence time dependent. And in order to boost and to enhance the efficiency of the system, we recommended a sequential nitrate recovery stage, which should be developed and incorporated into the modular reactor for optimal nutrient recovery. Thank you for listening.
Thank you, Professor Oladoja. Yes, great to see the results from that important work. Do we have any questions or comments? I don't see anything in the chat currently. Oh, here's a comment that came in. Just wondering, where was the modular reactor located to gather and process the urine?
It was a laboratory study. It was located in the lab. We conducted the experiment in the laboratory, the modular reactor. It was just a column reactor. You know, we used glass column reactor and packed with the reactive material, which is the thermally treated gastropod shell or raw gastropod shell.
Okay, great.